Welcome to Lecture 2 on biomolecules. In this lecture, we will explore the structure, function, and properties of 4 essential biomolecules: carbohydrates, lipids, proteins, and nucleic acids. We will also introduce enzymes and delve into their catalytic functions. 1. Carbohydrates. Carbohydrates are organic compounds composed of carbon, hydrogen, and oxygen, usually with a 2-to-1 ratio of hydrogen to oxygen. 1. They are one of the primary sources of energy for living organisms and play crucial roles in cellular processes. 1. Carbohydrates can be classified into 3 main types: monosaccharides, simple sugars; disaccharides, 2-linked monosaccharides; and polysaccharides, long chains of monosaccharides. 1. Monosaccharides. Examples include glucose, fructose, and galactose. 2. Disaccharides. Examples include sucrose, glucose plus fructose, and lactose, glucose plus galactose. 3. Polysaccharides. Examples include starch, energy storage in plants; glycogen, energy storage in animals; and cellulose, a structural component in plant cell walls. 2. Lipids. Lipids are a diverse group of biomolecules that are insoluble in water but soluble in nonpolar solvents. They serve various functions, including energy storage, insulation, and forming the structure of cell membranes. Common types of lipids include: 1. Fats, triglycerides, composed of glycerol and 3 fatty acids. 2. Phospholipids, essential components of cell membranes, consisting of a hydrophilic head and hydrophobic tails. 3. Steroids, such as cholesterol, which plays a role in cell membrane structure and acts as a precursor for other steroids like hormones. 4. 4. Proteins. Proteins are large biomolecules made up of amino acids linked together by peptide bonds. 4. They are involved in countless cellular functions, including catalyzing reactions, enzymes, transporting molecules, providing structural support, and serving as signaling molecules. 4. Proteins have 4 structural levels: primary, secondary, tertiary, and quaternary structures. 5. Enzymes are specialized proteins that act as biological catalysts, speeding up chemical reactions in cells without being consumed in the process. 5. They achieve this by lowering the activation energy required for a reaction to occur. 6. 6. Nucleic acids. Nucleic acids are macromolecules that store and transmit genetic information. 6. There are 2 primary types of nucleic acids. 7. Deoxyribonucleic acid, DNA, found in the cell nucleus. DNA contains the genetic instructions essential for the growth, development, functioning, and reproduction of all known living organisms. 8. Ribonucleic acid, RNA, plays a crucial role in protein synthesis by transferring the genetic information from DNA to ribosomes, where proteins are synthesized. Enzymes. Enzymes are biological catalysts that facilitate chemical reactions in living organisms. They are highly specific to their substrates, reactants, and can increase the reaction rate by lowering the activation energy required for the reaction to proceed. Enzymes are not consumed during the reactions and can be reused. Their names often end in -ase. Enzyme catalytic functions. Enzymes can act in various ways, including: 1. Lock and key model. The enzyme's active site is highly specific to the substrate, much like a lock fits a key. 1. The substrate binds to the active site, and the enzyme catalyzes the reaction. 2. Induced fit model. The enzyme's active site undergoes a conformational change upon substrate binding to improve the fit between the enzyme and the substrate, enhancing catalysis. 3. Cofactors and coenzymes. Some enzymes require non-protein molecules called cofactors or coenzymes to function properly. 3. They assist in the catalytic process. Enzymes are crucial for maintaining the biochemical processes in living organisms and play a fundamental role in metabolism. That concludes our lecture on biomolecules and enzymes. I hope you found it informative and gained a better understanding of these essential components of life. If you have any questions or need further clarification,